Hello students, welcome to EPG Pathsala. I am Dr. J. Arunachalam from Sastra University. Today we are going to talk on the module Introduction to Molecular Phylogeny from the paper Bioinformatics. The specific objectives of this module is to understand the architecture of phylogenetic trees, various tree construction methods, and validation of phylogenetic trees, and finally the applications of molecular phylogeny. All species in the earth are related to one another. This relationship can just be estimated in a quantitative manner. So we all know the fact that all the species are related to one another, which could be easily observed by looking at the species in itself. There are similarities which are progressing in a hierarchical way from an unicellular organism to multicellular organisms and organisms like human beings and other animals. So there is, when we observe that, we could see there is hierarchical progress in several levels, which eventually ends up in evolved organisms like humans or other animals. So not only having that or explaining that in a qualitative way, it is also possible to explain this relationship between the species in a very quantitative manner. A tree structure representing this relationship, which in a quantitative manner is called as a phylogenetic tree. In earlier studies, morphological characteristics were recorded from existing or fossilized species, which were used to obtain this quantitative relationship and which was represented by means of phylogenetic trees. With the revolution in sequencing technologies and development of efficient bioinformatics algorithms, this has allowed us to use DNA or protein sequences for phylogenetic analysis. So earlier, morphological traits were used for building phylogenetic trees, but with various bioinformatics methods and sequencing methodologies that we have here, we can use molecular information and describe similarities and dissimilarities between sequences in terms of phylogeny. So with a set of DNA or protein sequences from different species, we should be able to infer the phylogenetic relationship among the species. This follows an underlying assumption that the sequence considered has evolved from a common ancestral gene. Such set of sequences which comes out from a common ancestral gene are known as orthologous sequences. It is also possible that two sequences considered are similar because of some gene duplication events, and it could be present in the same species. Such sequences are called as paralogs. They might even have the same function, but they don't have a common ancestral origin. So they are called as paralogs. So phylogenetic informations are usually derived from an orthologous set of gene sequences, and they are called as operational taxonomic units. Architecture and features of phylogenetic trees. So this figure that is displayed in this slide shows an unrooted phylogenetic tree joining four operational taxonomic units. They are named as A, B, C and D. So a phylogenetic tree, as shown here, is an acyclic connected graph. The vertices of the graphs are called as the nodes and the edges are called as the branches. The nodes can be internal or external nodes. The nodes are connected by branches. The connection is in such a way that every node can be reached by a unique path from every other node. For example, from node A, we will be able to go to node D by means of a unique path. All phylogenetic trees are binary, meaning that any internal node can only split into two. So that could be seen in the figure also. There are two internal nodes and each of them splits into A and B, and the other node splits into C and D. So that is what we mean by saying all phylogenetic trees are binary. Each branch is associated with a certain amount of evolutionary divergence and is measured by an appropriate distance measure. Length of the branches corresponds to a certain amount of evolutionary distance. They are proportional to the evolutionary distance. Two types of phylogenetic trees can usually be constructed. They are the rooted and unrooted phylogenetic trees. A rooted and unrooted tree represents the phylogenetic relationship between sequences, but it cannot provide an evolutionary path. So the tree shown in one of the figures earlier was an unrooted tree. This tree showed us that the relationship between the species A, B, C and D, but it fails to tell us about the series of evolutionary events that led to the observed relationship. It just tells us the relationship. Similarly, it could also be noted that it is not possible to determine whether a given internal node is a common ancestor to any two external nodes in an unrooted tree. An unrooted tree can be rooted by using an outgroup, a sequence from a species which we clearly know as a distant homolog compared to all other species of our interest. So here this figure represents the 5 different ways of rooting a phylogenetic tree which was shown in figure 1. So earlier I showed an unrooted tree which is once again shown in this figure also, bordered by a rectangle. So that is an unrooted tree. If I want to root the tree, then if you want to root the tree, then there are 5 different ways of rooting this particular tree. So that is what is shown in figure. So rooting a tree allows us to trace correct evolutionary pathway. That is the application of why we should root a phylogenetic tree. So there are applications where unrooted trees can be used, and there are also applications where we need to root a phylogenetic tree. So it is not important that every phylogenetic tree has to be rooted. It depends on the application which we are going to subject our phylogenetic tree into. Inferred trees, gene trees and species trees. So a rooted gene tree constructed using 4 genes from human, chimpanzee, gorilla and orangutan is shown here. The tree was rooted using baboon sequence as an outgroup. So we have an outgroup here, and based on which the tree is rooted. This rooted tree can also be called as an inferred tree, as it depicts the evolutionary relationships that are inferred from our data. It may not be a true tree, which represents the actual evolutionary relationship. So that is a very important point that we have to remember. This is an inferred tree. There are statistical methods like bootstrapping, which can associate a confidence value to the inferred tree, signifying their resemblance to true trees. So there are validation methods such as bootstrapping. Relationship between gene trees and species trees. Even if the gene tree and the species tree look the same, we cannot assume the same time scales for gene diversion and speciation events. So that is what is explained in the figure. If 2 speciation events are close to each other, then a second mutation event can occur before the elimination of the first allele. So if that is the case, this can lead to difference between gene tree and a species tree. That is what is shown in the second figure. So you have a mutation event, and before the mutant allele is either selected or eliminated, we have a second mutation, after which a speciation process takes place. So now the result is we have different gene trees and species trees. So the sequences is not able to give enough information that correspond a gene tree with a species tree. So this difference is observed, it is very common in observing such difference is very common in molecular phylogeny. After understanding the properties of phylogenetic trees and the architecture of phylogenetic trees, it is important to know how to construct phylogenetic trees. So we will have to see the different tree construction methods. Phylogenetic tree reconstruction and subsequent analysis involves the following steps: an accurate multiple sequence alignment of orthologous sequences That is the input for phylogenetic tree reconstruction. And then we have to select an appropriate tree building methods. There are several methods that are used to build the trees, which we would see as we progress in this tutorial. So the next step is to use an appropriate tree building method for our data, that is the multiple sequence alignment. And then the most important step, the most important final step is to assess the accuracy of the reconstructed tree. Any tree that we have constructed should be validated with proper validation procedures. Molecular clocks can be set to estimate the time of divergence of ancestral sequences, but that is optional. So sequence alignment and phylogeny, that an accurate sequence alignment always leads to an accurate phylogeny inference. So multiple sequence alignment, commonly known as MSA, is a primary requirement for reconstructing a phylogenetic tree. Every column in a multiple sequence alignment is considered as a site of independent evolution. So that is something important that we have to understand. So multiple sequence alignments are present as columns. You can consider multiple sequence alignments as several columns, and every column in a multiple sequence alignment is considered as a site of independent evolution and is assumed to be evolving as per the observed sequence variation in that column. That is also important. It is assumed that these sequences are evolving as per the observed sequence variation in that particular column. So gaps are always ignored during phylogenetic reconstruction. Both protein sequences and DNA sequences can be considered to build phylogenetic trees. Nucleotide sequences of a pair of homologous genes are statistically more informative. Sequences should definitely be homologs, and MSA made of non-homologous sequences will also produce a tree. So that is a very important point that you have to remember. The tree reconstruction programs can take any input and it can produce any tree. So any MSA or multiple sequence alignment can produce a tree. So even if I have a multiple sequence alignment of non-homologous sequences, which is completely meaningless, the tree building programs will produce a tree. But the question that we have to ask is such a tree correct? It is going to be inaccurate, it is going to be incorrect, because the multiple sequence alignment that we gave as an input is for non-homologous sequences. So such a tree is incorrect and it is also biologically irrelevant. Explaining the steps involved in building phylogenetic trees, we move on to different tree building methods. An accurate multiple sequence alignment should be converted to numerical data that is necessary in order to construct a tree. So we have the multiple sequence alignment as an input. Now the next step is to convert that multiple sequence alignment to appropriate numerical data. Based on the numerical data, we will be able to construct a phylogenetic tree. There are three broad classes of methods which are used to build phylogenetic trees from any given multiple sequence alignment. The first of them is the distance based methods, the second is the maximum parsimony method, and the third is the maximum likelihood method. Now, even though we have these three different types of methods, there is no single best method for building a phylogenetic tree. We cannot recommend always that this method works best always and this method doesn't work well always. It is case dependent. In some cases, distance based methods work well. In some cases, maximum likelihood method work well. They have their own advantages and their own disadvantages. So there is no single best method for building a phylogenetic tree, and the accuracies of this method stated are case and data dependent. A guideline for selecting an appropriate phylogenetic tree building method. We saw that primarily there are three different methods based on which phylogenetic trees could be built, and this flowchart can approximately guide us to select the most appropriate method. There is no single method, single procedure that we can say that it could produce the most accurate phylogenetic tree, but this is a general pipeline that could be followed. So choose a set of related sequences. We have to obtain the multiple sequence alignment. Is there a strong sequence similarity in the alignment? If yes, we can go for maximum parsimony based methods. If the answer is no, then the next question that we have to ask is there a clearly recognizable sequence similarity? If there is a clearly recognizable sequence similarity in the multiple sequence alignment, then we can use any of the distance based methods. If there is no clearly recognizable sequence similarity, then we can use the maximum likelihood based methods. After using maximum likelihood based method, we analyze how well the data support the prediction. That should always be done. Same for maximum parsimony method as well. So by this approximate guideline, we will be able to select an appropriate tree building method for our sequence data. Distance based method, modified Jukes-cantor. The multiple sequence alignment that is shown as an example, this slide has 25 columns and hence there are 25 sides here. A similarity matrix can be generated based on above alignment. For example, sequence A and sequence B contains 21 matches out of 25. It is shown below. Therefore, the similarity between sequence A and sequence B is 0.84, since 21 divided by 24 is 0.84. So, sequence A and B and the corresponding alignment is shown below. This is a continuation of the distance based method. What we saw earlier, how to generate similarity matrix, how to generate a similarity score between two given pair of sequence in a multiple sequence alignment. A matrix can be generated by calculating the similarity between all pairs of sequences in a multiple sequence alignment. So that is what is shown in this slide. So the same similarity indexes can be calculated based on substitution matrices such as PAM or Blossom matrix. The example shown here is for gene sequences, but once again the same could be done for protein sequences also. So in the alignment we had 5 sequences, we named it A, B, C, D and E, and this corresponding matrix gives us the similarity between the sequences. For example, the similarity index between sequence A and E is 0.52. Similarly, sequence B and E is 0.52 as well. So this is in continuation with the previous explanation for a distance based method. So earlier what we did was we created a matrix based on similarity. We created a similarity matrix for a given multiple sequence alignment. So the second step is to convert this similarity scores to corresponding evolutionary distances. Consider a situation given below. When an ancestral sequence mutates into two contemporary OTUs. So ancestral sequence A, T, G and T, and then it mutates to AGGT, which is observed. Similarly, AC, G and T. So in second position, T mutates to G in one case and mutates to C in the other case. So you have two sequences now. This is what we observe right now. We only have to infer the ancestral sequence. So this is the observed sequence and we make an alignment based on the observed sequences. This alignment shows only one mismatch in nucleotides at position 2. However, two nucleotide substitution can have actually occurred. The similarity in the first position AA might also be a reverse mutation. That is, during the course of evolution, the first position might have changed from A to T and from T to G and then from G to A. So using a similarity matrix alone will underestimate the evolutionary distance. So what we observe is A in both the cases, but there could have been intermediary mutations in the first position and there could have been a reverse mutation where once again changed to A when we observed. So this cannot be treated to be the same as the mutation that we saw in the second position. So a similarity matrix using similarity indices alone will underestimate the evolutionary distances. So we have to make appropriate models that estimates the evolutionary distance more accurately. Jukes-cantor substitution model. So Jukes-cantor substitution model, in this model the percentage of mutation is not equal to the observed dissimilarity in the alignment. It assumes equal substitution probabilities for all bases, that is 25%, and models the difference in sequence against the evolutionary distance. So that is what is shown in the graph. So similarity index and its corresponding evolutionary distance. So for example, from our similarity matrix, if we have a similarity index of 0.84, it shows a corresponding evolutionary distance of 0.14. Similarly, a similarity index of 0.77 will show a corresponding evolutionary distance of 0.52. So this model can be used to convert similarity scores to evolutionary distance. This Jukes-cantor substitution model is one of the very simplest model that can be used for calculating the evolutionary distances. There are several other models like Kimura's model, etc. Several other models based on which the evolutionary distances can be estimated from the similarity indexes. Distance based method modified Jukes-cantor. So the similarity score, as explained in the previous slide, 0.84 between sequences A and B in our example becomes 0.18 in terms of evolutionary distance. Similarly, the evolutionary distance matrix can be constructed from the similarity matrix. Neighbor joining method for building a tree from distance matrix. Neighbor joining methods uses the distances, evolutionary distances that have been estimated by the method that were explained earlier. So it starts with two sequences which are separated by a line which is equal or proportional in length to the evolutionary distance between the sequences. For example, in this case, A and B. The next sequence C is added to the tree such that the distances between A, B and C are appropriately equal to the evolutionary distances. The fitting is done by least square method. Similarly, the sequences are added one by one and the branch lengths are fitted appropriately by least squares method. So for our example sequences, if we calculate the distance based on Jukes-cantor method and build a neighbor joining tree, this is the tree that we would get, which is shown in the figure. Distance between any two sequences is equal to the sum of the length of the line segments joining them. We call this type of tree as additive trees. An UPGMA method could also be used, whereas it is inconsistent in the case of data that is not ultrametric. A phylogenetic tree could also be visualized as a dendrogram, which is shown above in this particular slide. A dendrogram shows evolutionary distances along the horizontal axis and assumes a root somewhere in the middle of the tree. The branch connecting sequence E to the rest of the tree is assumed to be the root in our particular case. The next method for tree reconstruction is the maximum parsimony method. In the neighborhood joining method, which we saw earlier, the information about ancestral identities at each position in the multiple sequence alignment is lost. What we have in the neighbor joining method is the overall sequence similarity and the corresponding inferred distance from that sequence similarity. So in such a setup, the role of individual sites in my multiple sequence alignment is lost. That can be used effectively in parsimony methods. So in the NJ method, information about ancestral identities at each position in a multiple sequence alignment is lost. Maximum parsimony method assumes that evolution follows the shortest possible route and the correct phylogenetic tree is therefore the one that requires minimum number of nucleotide changes to produce the observed differences. The columns in a multiple sequence alignment are considered to be an informative site. So each column in a multiple sequence alignment can be considered as a site of evolution, but each column need not be informative. So among the columns in a multiple sequence alignment, we have to select the columns that have necessary information for us to construct a phylogenetic tree. Only such columns are called as informative sites. So how do we select those columns? Those columns are selected only if it has at least two similar characters. So we have put a threshold now and say that in a column of a multiple sequence alignment, if it has at least two similar characters, then we call that as an informative site. The analysis is done for each possible informative site alone, and an overall tree with minimum number of substitution can be identified. This method, however, is computationally very demanding for large number of sequences. Maximum parsimony method for tree reconstruction. For our particular example, which is used to demonstrate the maximum parsimony method, leaves columns 3, 5, 7 and 9 as informative sites for our MSA. So the figure also shows the three possible unrooted trees along with the number of substitution for site 5. The tree 1 has minimum substitution and is selected. This is repeated over every informative site of the alignment, and an overall tree with minimum number of substitution can be identified. This overall tree is called as the maximum parsimony tree. The third method for phylogenetic tree reconstruction is the maximum likelihood method. Commonly it is known as the ML method. ML method uses, it is also like maximum parsimony. It uses each position in an alignment and evaluates all possible trees. So like a neighborhood joining method or distance based method, it doesn't operate only on the distances which is obtained from the similarity scores. So ML method also uses each position in an alignment and evaluate all possible trees. So it calculates a likelihood for each tree and then seeks the one with the maximum likelihood. So all possible trees are constructed and the likelihood values for each tree is estimated, and the tree which contains the tree which is estimated to be maximum likelihood is considered as the best tree. So at each site the likelihood is determined by evaluating the evolutionary model. So we need to have some evolutionary model. It could be Jukes-cantor model, Kimura model, or more advanced models. So based on the evolutionary model, the evolutionary models are used to generate the observed data. The likelihoods for each site are then multiplied to provide likelihood for each tree. So you calculate based on the models which I explained earlier the likelihoods for each site, and then they are multiplied to provide the likelihood for each tree. Maximum likelihood method is the slowest and most computationally intensive method, though it seems to give the best results and it is always the most accurate tree. But the disadvantage, as I have mentioned, is that it is a slow method and it is computationally intensive. However, it produces the most informative tree. Comparison between tree building methods. So this table compares distance based methods, maximum parsimony method, maximum likelihood method. You see that the distance based method uses only the pairwise distances, whereas the maximum parsimony method, it uses only the shared derived characters, that is the informative sites alone, whereas the maximum likelihood method uses all the data. Similarly, the maximum likelihood method, it maximizes the tree likelihood given specific parameters values, but it is very slow. Maximum likelihood methods are very slow, whereas distance based methods are very fast methods. Similarly, maximum likelihood method is good for very small data sets and for testing trees built using other methods. That could be trees that are built using other methods can also be tested by means of the maximum likelihood procedure. Whereas for a distance based method, it is very fast as well as large number of sequences can be used in a distance based method. Validating phylogenetic trees, the bootstrap method. So after building a tree, it is important to validate the phylogenetic tree. So we need to know how well build the tree. There should be some quality metrics that should tell us how good a molecular phylogeny, how a tree has been built by us. A new alignment of the same length is generated by randomly selecting columns from the original multiple sequence alignment with replacement. So what we have to do is, we had original multiple sequence alignment based on which we created the tree. Now we have to randomly resample with replacement and produce several sets of multiple sequence alignment, and we also have to produce corresponding trees from the random alignments that we have generated. So we have a few thousand random trees, and we also have our original tree from the original multiple sequence alignment. Now what we can do is, the frequency with which a chosen phylogenetic feature of the original tree, a branch for example, so that particular feature we have to check how many times it reappears in the bootstrap population of the trees, and it is estimated to be the confidence associated with that feature. So by means of this bootstrapping, we will be able to associate confidence measure to that particular phylogenetic feature. How to do bootstrapping procedure earlier? This slide shows a tree which has been generated and has been validated by means of bootstrapping procedure. So we see that every branch is associated with an appropriate confidence value. We see that there are branches, for example, branch which differentiate the human gorilla has a 99% confidence value. Similarly, there are also branches, the features of phylogenetic trees, there are also branches which has confidence value which is lesser than 50. So such branches can be termed as less significant branches, whereas the branches that are above a particular threshold can be considered to be significant branches. A phylogenetic tree, this phylogenetic tree was drawn based on the NADH dehydrogenase sequences and its associated bootstrap values. Applications of molecular phylogeny methods. Molecular phylogeny methods has several applications. Here I have listed quite a few of them. The first application that we could talk about, HIV and the Florida dental case, where a Florida dentist was suspected to transmitting HIV infection accidentally to his patients. So some of his HIV positive patients had also acquired infection through some other means, and the exact number of it needed to be determined. So phylogenetic tree was built based on the GP120 sequence of the HIV, and it shows that phylogenetic tree is shown in the figure, and you could see that there is a clear clade of HIV infection that all groups together which are transmitted through the dentist. You have to see that GP120 gene mutate very fast, and there could be several variations of GP120 sequences among the HIV affected patient samples. Phylogenetic tree built based on that gives a clear separate dental clade which is named as the dental clade, and such patients were all grouped clearly together in the phylogenetic tree. So this was able to answer the exact number of people to whom the HIV infection was transmitted through the Florida dentist. So phylogenetic trees were also used to identify the origin of cellular organelles. So this example, this tree is a phylogenetic tree built based on mitochondrial and chloroplast DNA sequence. Clearly shows that both of them are endosymbiotic organelles, but mitochondria is much closely related to alpha purple bacteria, whereas the chloroplast closely related to the cyanobacteria. So probably chloroplasts evolved from cyanobacteria through endosymbiosis, and mitochondria evolved from alpha purple bacteria through endosymbiosis. The archaeal diversity. So the tree that has been shown here is the universal phylogenetic tree. The phylogenetic trees of rRNA sequences belonging to organisms of Yellowstone National Park showed an extended monophyly of archaea. The data also showed, the data on the corresponding phylogenetic tree also showed the relationship between marine archaea and high temperature archaea. The tree was inferred by using maximum likelihood method with 1620 homologous sites from all the organisms that has been considered. So you could see that clearly there is entire grouping of bacteria, archaea and eukarya. applications of molecular phylogeny methods. So, identification of selection pressures and surveillance of HIV. The selection pressure on any particular branches or clades of a phylogenetic tree can be identified by the ratio of synonymous to non-synonymous substitution in codons. The D/DS ratio being greater than 1 signifies a positive selection. The study conducted by Yoshida et al. In 2011 reveals very important aspects of HIV infection among Japanese population. Identify the selection pressures on the phylogenetic tree built using ENV gene sequence from 16 samples collected in 1980 and 2000. This result showed that the selection pressure was weaker in 2000 than in 1980s. This indicates that it is easier for HIV to infect a new host in 2000 than it was 20 years ago. In other words, this phylogenetic study has shown us that HIV is becoming more virulent. At least it is more virulent in 2000 than it was in 1980. After describing the phylogenetic tree, the construction methods, and what we have to do is we have to look at the possible problems and errors in a phylogenetic analysis. First of all, small inadequate data or limited sampling will cause errors in phylogeny. What we quite often see is that we don't bother to see whether we have adequate data to infer phylogeny. Most often people would just try to infer phylogeny with inadequate data, which is always going to give an error in a tree or an incorrect tree. Similarly, sequences undergoing rapid nucleotide changes or very fast changes, this might skew the results of our analysis. So, such sequences which are mutating very fast can also be a potential problem leading to error in phylogeny analysis. Similarly, mutations involving duplication, deletion, insertion, etc. also give inaccurate signals. Genomic hotspots are small regions of genome with rapid evolution. Such regions are not easily detected. Similarly, homoplasy or nucleotide changes that are similar but occurred independently over separate lineages are often identified as inheritance. So students, let's now summarize what we have learned in this module. The tree structure, which represents the relationship between different species in an evolutionary perspective, is known as a phylogenetic tree. The trees could be rooted or unrooted. A tree is normally rooted using an outgroup sequence. The inferred phylogenetic tree need not be the same as a species tree, but it is usually a close approximation of the species tree. The trees could be built by distance-based methods, maximum parsimony method, or maximum likelihood method. We have to select the most appropriate method for our data, considering the advantages and disadvantages of these methods. And the built trees should always be validated by a procedure like a bootstrap procedure. The phylogenetic methods have a wide variety of application like monitoring disease epidemiology, species conservation, pathogen surveillance, identifying origin of cellular organelles, etc. Thank you.